In this trikinetic mobility assay, a DAM2 activity monitor will be used to measure Drosophila's activity. Every time a fly passes across the infrared beam, it activates the counter which measures the number of times a fly passes during a set period of time. To begin, flies will be sorted by sex and placed into individual tubes with a small amount of food in them. The tubes will then be placed into the DAM2 system, which will then be connected to a computer for activity monitoring and data collection.
To begin the experiment, food was prepared for the flies using 8 ml of tap water with 2 grams of instant fly food medium. Using prepared food, individual tubes used for the DAM2 trikinetic mobility assay were pushed into the vial of food to accumulate a small amount at the bottom of each assay tube. Each tube was then capped to ensure the food remained inside. It is important to have a small amount of food at the bottom of the tube because it provides nutrients to the flies while their activity is being measured throughout the assay. To gather flies for the mobility assay, 20-day aged flies were passed from their vial to an empty one. Once passed with the use of a funnel, FlyNap, the most common fly anesthesia agent, was placed into the vial. After a few minutes, the Drosophila enter a temporary sleep.
The prepared tubes with sleeping flies inside were carefully placed into the DAM2 Drosophila activity monitor. The tubes were slid into the holes of the apparatus and centered so that the laser beam would run down the middle of the tube. Rubber bands were used and wrapped around the tubes so that they stayed in place. Once the apparatus was prepared, it was taken to the fly incubator where it was plugged into the power supply interface unit. The fly incubator is temperature controlled and has a 12-hour on, 12-hour off light cycle, which controls the circadian rhythm of the flies. A power brick with an adapter was plugged into a standard outlet and connected to the power supply interface unit to supply the power. A third wire from the power supply interface unit goes into the computer to collect the data. On this screen, you can see that there is a green box with a 1. This means that the monitor is connected correctly and it is actively collecting data. At the top, you can see that it says next reading in 1 minute. On this screen, you can see that the reading interval is at 5 minutes. This setting can be changed, but it was kept at 5 minutes for this experiment. Again, from this page, you can see when the next reading is. In this page, you can see the current data being collected.
Once activity monitoring was complete, collection would be stopped on the computer and data would be saved on a USB. The computer would then be shut down and the tubes would be emptied, cleaned, and autoclaved. The data on the USB would then be analyzed. So in this example, we're going to be looking at 100 minutes of activity for flies. And this is just some sample data. And as we can see, that the flies were still on FlyNap for the first 5 to 10 minutes. So there's no movement detected. And then we can start to see that each fly has a little more movement or less movement than the other. So we take the average of the females, which are 1 through 4, and the males, which are 5 through 8, and we use this to plot our graph in MATLAB. So in MATLAB, we're going to import this Excel document into our MATLAB by using import data, and the document is just called example. We have our example here, and as we can see, we see all of our cells, but we're only interested in the average and time, which is our independent variable. So what we do is we click and highlight the rows, the columns that we want. So now we're going to go to the output type and we're looking for column vectors. And we're going to import the data into MATLAB. So now it has our variables, but we want to create a function so that way we can create a plot. So generate function, and now we have a script. And we can scroll down and we can see our function, but what we're really interested in is the variables because that's how we'll create our plot. So here we have time, average female, and average male. There's numbers behind it because I ran this a couple of times, but the first trial would be time, average female, and average male. So we're going to have 2 plots because average male and average female will be on different plots. So we'll indicate plot figure 1 first. And then we're going to have plot of time 4 on our x and average 3 on the y. And we're going to add in some labels. Now we can see our figure, but what we want to do is find the area under the curve. So to do so, we're going to plug in area and our x and y. And then we're going to use trap C. That's the command to solve for the area under the curve. So we identified the area that we're calculating with area, and then trap C will calculate. And so we will plug in the same time 4, average female 3. And there we have our area under the curve. So now we have that. We're going to then plot our average male. So that'll be figure 2. Here we have our plot. We would put in X label and Y label and title as well, but for now we'll just get our numbers to compare. So we have area of time 4 and average male 2, not 3. We identified our area that we're calculating, and we'll use that same trap Z as our command. And time 4 and average male 2. And now we can compare with 406.7188 versus females activity, which is 472.1875. So our numbers can help us identify that the females are more active than the males.